Hello, I am Brynn, and I'm giving a talk about Intro to Bioinformatics. To start off, so I am Brynn. A little background about me. I am a student at the University of Pennsylvania. I am a senior at this point. I am studying computational biology in the School of Engineering. So that pretty much means I take a lot of computer science classes and a fair amount of chemistry and biology and statistics and a lot of upper level biology and genetics. I am also a hacker. I go to a lot of hackathons, and I was the director of the PennApps hackathon, if you've heard of it, like last spring and the fall before that. And I also participated last time. So yeah, here we go. So this talk's a little bit different than what you've probably seen today if you've been paying attention. This isn't a language or a type of technology that I'm teaching you about. This is kind of a more general topic, so I'm kind of going to do an overview. I'm assuming most people here have a background in either biology or computer science. They maybe know a little. And I'm going to start by just telling you what bioinformatics is and why it's cool, and maybe, even some downfalls of it and how to fix some problems that you might run into. And then I'm going to give some really cool examples of things that you could actually do on your own to do your own computational analysis of biology information. Okay, so what is bioinformatics? Very simply put, it is biological problems that you solve with computational solutions. So there's kind of a process to any bioinformatics solution to anything you might be doing. The first one is to pick a biological problem. Not necessarily always the first step, but generally a good place to start. I've listed a couple of things that are wider topics in biology that I'm actually gonna give you some examples of later. Genetics, so that's studying DNA. Evolution, so how different organisms have evolved from each other. Proteins, so these little tiny molecules that actually make up everything in your body. And medical image analysis. Good, so step 2 is usually find some data. So bioinformatics is really quite frequently a kind of big data approach to biology problems. So yeah, there's a lot of data, and it's kind of a struggle. If you've ever thought about something like Facebook has a lot of data to go through, biology has way more data to go through. The scale is just unbelievable. In every single cell in your body, you have millions of base pairs of DNA. So if you're trying to at all keep a resource bank of different people and the different maybe their genomes, several different people and all the genomic information they have, it's actually millions and millions of base pairs, millions and millions of data points. And think about something like financial transactions. So a bank has to keep track of people using their credit cards every day and their debit cards every day and cashing checks every day, and this is millions of people doing this. So something of that scale, you kind of got to think on that scale. So you can't really solve the thing. You really need to think a lot about how you're storing your data and think a lot about algorithms. So what's the most efficient way to parse data and get information out of it? I'll give a good example of this later. So there are actually a lot of databases online that already came up with very elegant ways to store all of this crazy amount of data, and they have it available to the public, which is kind of crazy if you think about it. You can actually see the genome of different organisms online and play with it, and it's totally for free. And I'm going to go through an example of this later too, playing with one of these databases. So The next step in the process is coming up with some kind of computational approach for analyzing your data. And yeah, there's a lot of them. I listed a few here. If you're in computer science, these probably sound like buzzwords to you. So one thing you can do is a machine learning approach. So machine learning, usually when you use machine learning, you're trying to classify things into categories. So maybe, you have information about genes and you're trying to figure out like which organism they belong to or which type of functional group they belong to. And if they look similar, they might do one function. And if they look very, very different, maybe they're from an older organism or they have some kind of different function. So machine learning can be useful for something like that. Graph theory. So when you're looking at genes and how they, essentially how the proteins interact that the genes create, it kind of forms a really complicated network. And you can actually use graph theory to apply to that network of, let's say protein A interacts with protein B and then interacts with protein C and D and E, and there's some very long chain, but protein X interacts with all of them. You could use a graph theory approach. Like a very basic thing would be to say, oh, well, put it on a graph so that every protein is its own node on the graph and look at all the connections between them and say, oh, it looks like protein X has a lot of connections, so it's probably really important. That's a pretty basic graph theory approach, and you could extend that out and use all of these really complicated graph theory algorithms to look at this network and figure cool things out about it. And again, it's just normal computer science approaches with just biology data. Natural language processing. So I've actually used this before to look at paragraphs describing what genes and proteins do and look for keywords and compare between different genes and different organisms and see what the differences are and try to get information out of this. Again, which genes are important, maybe which genes belong to a group based on the description of what they do or what they interact with. And dynamic programming is actually used a lot, if you've ever heard of this. If not, again, it's totally fine. It's used a lot for looking at sequences because, again, analyzing sequences is actually, sequences of DNA, is actually very difficult and uses a lot of computational power. So you need to be very smart about it. Another common computational approach, both for looking at your results and also sometimes as part of your actual analysis, is using visualizations. So the bottom one here is protein structures. So, there's some data that says, oh, this part of a protein looks like this, or it's a different type of element, a different type of sub-element, and showing how they interact in different circumstances and how these proteins can come together. Because proteins are so tiny, we actually can't do that at all in real life or really see it. So we need computers to actually see it and make it really big for us so we can actually understand what they look like at that very, very tiny scale that we could never actually visualize. And up top, I have a heat map. So this is showing how different genes act in different circumstances. So let's say one gene, let's say there's an experiment that puts in a molecule, something like glucose, which is sugar, and looks at how different proteins are expressed more or less based on putting in the sugar, this extra molecule. And we can actually visualize how they interact on a wide scale with lots of different genes and in lots of different experiments with heat maps. And it makes it a lot easier just to look at it and see what general patterns are. And then you can even apply computational approaches on top of that and learn things about these heat maps and learn about how these genes interact and how they're expressed and how they react to different elements. And the last step is verify, verify, verify. This is very heavily into statistics. Because when you're doing computational approaches, you aren't actually proving anything straight out. You're generally just making inferences based on lots and lots of data that you've looked at, and you usually say, well, since I looked at all of this ridiculous amount of data, it makes sense that what I found is important, and then you cite some statistical report. You say, it's statistics. If you've taken a statistics class, you say it's statistically significant based on X, Y, Z. And this is extremely important because, again, it's very difficult to actually verify things with computational approaches. So we have to just prove that what we're doing is very, very likely to be true. Okay, so there are a lot of shortfalls to bioinformatics, and it takes very, very smart people to try and get around these problems. The first one is just that normally when you're creating your machine learning models or something like this, you kind of have to create cutoffs on your own. You kind of say, I'm pretty sure this belongs to this category if it is this much similar to whatever the category is. And that's not very certain, and you just kind of have to have a lot of experience to be able to actually draw those cutoffs and make it actually make sense. And again, there's way too much data for anybody to ever even comprehend. So you have to be very smart about how you store it and how you analyze it. And the main problem that I'm seeing right now is just that there's so many different tools, and there's all of these different file types for saving information about genes. Even something like that, just there's no standard format for saving information about genes. There's tons and tons of different file types. And a lot of what bioinformaticists do, unfortunately, at this point, is convert file types or convert your data so that it'll fit into some tool that somebody made to visualize your data or to analyze it or do something else. Okay, let's go through some cool examples. There's a lot of examples with genomics. You could use bioinformatics approaches for pretty much everything in genomics. I'm going to explain a little bit about what it is for those of you who maybe haven't taken a biology class in a while or haven't ever taken one. So DNA, starting really basically, is just a string of the letters A, C, T, and G. That's all you need to think about it as a bioinformaticist, as somebody who's going to just be reading strings, which, if you're a computer scientist, you're already starting to think oh, how can I analyze this as a string, and what ways can I compare strings and learn things about it? And again, we have millions of them, and each little part, so maybe 100 of these A, C, T, and G strings is called a gene, and that's a functional group. So we're actually going to look at a gene that's a few hundred base pairs. Again, that's what an A, C, T, or G is called. And it's the insulin gene, so it creates this molecule that's very important to the disease diabetes. And it is the actual DNA in all of your cells that codes for this molecule to be created. We have something in genetics called the central dogma, and it's very basically the idea that you take this DNA that's stored in all your cells and you use it to make proteins, because DNA can't actually do anything, it's just information. You can almost think of it as just binary, zeros and ones. You can't actually do anything with that. You need to kind of translate that and turn it into something that's actually useful. So proteins are the things that are actually useful. They're the things that actually interact in your cells and build things in your cells and do things. And there's weird research that proves that sometimes you can usurp this or go backwards, but this is the central dogma that we will assume for now. Okay, so let's take a look at some resources. One of the best resources is NCBI. So this is kind of the National Institute of Health, the United States government resource of tons of genetic information about every kind of organism you could ever imagine, and all sorts of information you could never even begin to go through. So this page is all the information they have on this gene insulin. It's in Homo sapiens, so it's a human gene. Again, there's a lot of stuff here. They have lots of cool visualizations. But a lot of the things on the government websites are kind of outdated in terms of visualizations. But yeah, so there's a lot of information here. I wanted to show off, you can actually, on this page, you can get the actual A's, T's, C's, and G's that make up this gene and do cool things with it. So again, it's kind of up to you to come up with the questions that you have and the things you want to solve. But they have a resource that's very important and that a lot of people use called BLAST. And BLAST is, let's say you're a biologist and you come up with a list, you find a gene in an organism and you don't know what it is or what it does. You can actually search just the pure A, C, T, and G sequence and figure out which organism it belongs to and what its function is. I've also used it to compare between organisms. So maybe I'll have a gene in one organism and I'm looking at something really closely related and I want to kind of tell the difference, see how related organism A and B are and see how related A and C are. So I will look at a particular gene or a very important gene and I'll use this tool called BLAST and look at the exact sequence and how closely it matches to the same gene in this other organism. And this is the kind of result that you get when you BLAST. So I just took this gene that we were looking at here and put it in this BLAST algorithm. And there's a couple different BLAST algorithms, but the one that they use on the website is pretty good. And it just shows you the alignment to everything in its database, and it does this very, very quickly. So this one actually has an exact match because I stole it from the website, and of course it will have an exact match. It matches to the one that I took it from. And you can, again, if you're looking at different types of genes or different types of organisms, it's actually very useful to see how similar these genes are. And maybe you can learn things about the evolutionary tree, what organism evolved from what this way, or if you have a gene on your own, you can figure out where it belongs or what it's supposed to do. So BLAST is a really cool resource. There are lots of other resources, and if you are a programmer, you probably want to access their API. I use this a lot. So all of these resources that NCBI has are available through their API, and you can call it and get all the data by yourself. I had a couple examples here. So this is an example. You just start with their base sequence, and you do one of their search functions, get a summary or fetch or do something like that. You pick a database that you want to query. You pick the ID of the gene and the type of information you want to return. And this is returning XML. So again, if you're doing a computational approach on your own, you can go through their API and get all of this information for whatever genes or articles or anything you want all on your own. And you can analyze them on a larger scale. And that's actually pretty, again, you have to pose your own questions and figure out what exactly you want to be searching. But it's actually a pretty cool thing to be able to search all of this information pretty instantly. Let's see. Yeah. So again, you can return different file types and you have cool things you can parse. This is actually this is an example of an abstract of an article. So this is another cool thing. It's not just the pure string ACTG that it has online. It actually has every article that's ever been written about biology and genetics, and it's all online for you to access. I actually use this to do natural language processing on the abstracts to find information, as I said before. So this is very useful because again, I can just query the API for all of the articles that I want to find, or I can start by doing my own query, getting all of the article names or numbers that I want, and then getting a query and taking out these abstracts and doing some kind of processing on them. And yeah, that's one very interesting approach to doing this. And there's tons of other things you can do with genetics. Again, I'm not really going to go into everything because that would take forever. And I love genetics, I'm a geneticist, so I have lots of examples. But I wanted to show you some other interesting things you can do with bioinformatics other than just look at these 4 characters and learn things about them. So evolution, again, I talked a little bit about this before. It's actually a very difficult question that we can't really solve. We kind of, like in theory, you can look at just purely a string, like all the DNA in every organism, and compare them next to each other and say, well, this one is more similar to, organism A is more similar to organism B, so they're clearly more related and they belong together on the greater evolutionary tree of life. But it's actually much more complicated than that, and there are a lot of like weird biological problems that lead to weirder computational problems. For example, genes that are crucial to functioning are very rarely changed. So, maybe organism A and organism B overall look really similar in terms of their DNA, but actually organism C it has the exact same like crucial function genes as organism A, and it's just some of extra DNA that's not actually useful that's been changed significantly. So those 2 organisms are actually probably more related than A and B. And this makes making trees really complicated and figuring out the evolutionary story really complicated. You can also incorporate data about how organisms look, or like how long their spines are, or et cetera, et cetera. And the more information you put in, the harder it is to process all of it and learn things about it. Protein structure. So again, I showed a picture before. You can do a lot of cool like visualization and 3D modeling to try and figure out how proteins interact. Because proteins actually like physically lock into each other. I think they call it like a lock and key model, but we can't see it, so we kind of have to create these computational models to figure out how they work. And again, you can think of protein interactions as a graph and analyze it with graph theory and do some cool things there. Another thing you can do is medical image analysis. So I actually, somebody I know had this problem and was having some trouble. So she was actually writing like Java code to analyze this image. And it wasn't this one in particular, but the whole point was she wanted to be able to find a tumor in like an MRI image. And she broke it down into a bitmap. So she looked at like the color of every single pixel. And she essentially wanted to be able to circle the tumor. So there's a lot of computational things involved there. She ended up using some algorithm that like goes through every pixel and just looks for like groupings of things that are the same color, and this particular like extremely light color that shows up in an MRI as a tumor. And doing this sort of thing makes it way faster for for patients to get analyses of their MRIs instead of waiting for doctors. It's always good to have a professional look at things, but sometimes it takes a lot of time and you really can't afford to wait. So there's a lot of really cool things, kind of more in the medical space you can do too. It's definitely not just limited to genetics and all of these like tiny things or theoretical things. It's things that are actually very applicable and can be solved. And bioinformatics is really just any biological problem that you can solve using these computational approaches. And that is it. If you have any questions for me, you could always tweet me at @bclaypoole, and I'm more than happy to answer. And I think we can have a question and answer session right now too.
You can. So the first question from the audience is, how did you get into bio and CS as a combo?
That's a really good question. So I was actually very into biology when I was younger, and I just took like normal biology in school and really loved it and started studying it on my own and like going to, very cool biology summer camps, and because I'm a really cool kid, I always have been. So I've really always been interested in that. And when I was in high school one summer, I signed up for a program, like a free summer, again, one of these free summer camps. It's called Summer Ventures, and it's like in North Carolina, because I'm from North Carolina. And I actually signed up for a class called visualization that I thought was about like seeing in the eye and like biology things. and it was actually about computer science. And I knew nothing about computer science, so I went and I met a lot of cool people who taught me a lot of really cool things, and I started really getting into computer science. And I couldn't really choose between biology and computer science, so I ended up just finding something that allows me to do both.
That's awesome. I think we have time for one or two more. So we spend a lot of time thinking about how to make our community more inclusive to like people who are traditionally underrepresented in tech, but theoretically like they know CS already. How do we make like hackathons and hacker culture more accessible to people who are not already programmers, who might like be in fields like biology and or whatever and have interest in this stuff?
Yeah, that's a great question. So I actually work on this as part of PennApps too. I started something called PennApps Health. I also started something called StitchFest, which is in the similar vein. But we essentially have these like kind of subgroups within our hackathon that are completely devoted to these fields that are not just straight up computer science or hardware. With hardware, we're starting to pull in people who are more into like electrical engineering, and that's great. But there are tons of people who have lots of other interests that aren't necessarily those in particular. So I think it's really nice to kind of create these sub-communities and try to have this focus of like, here's a group of people who are just going to focus on, healthcare hacking, like things that can help the greater healthcare industry. And those people, some of them were doctors that I actually I taught like a summer class on Node to try and like help them get integrated, and it was really awesome. And we actually managed to pull in some people from medical school. So, I think this is like a very interesting problem and something we can really work on a lot by trying to say, we know you guys have other, we know you have other interests, but we really, really like this idea of like constantly building new things and trying to like come up with solutions to these big problems. And even though we don't understand these problems, we would love for you to come to our event and learn about how we operate, and for you to teach us things about the problems in your world, so we can try to solve them together.
Awesome. Any closing thoughts, and how can people get in contact with you going forward?
Yeah. I don't know about closing thoughts. Again, I highly encourage anybody who thought this was cool to just kind of play around with some of these resources. It's a little difficult because biology, like there's so much biology terminology, but something like looking at a string of letters is something that computer science people understand pretty well. So, I encourage you to just play around and look at what some of these databases have to offer, especially the ones that I had listed earlier. And if you want to reach out to me, again, I have my Twitter handle up here. You're more than welcome to tweet at me. If you want to email me, you can try Brynn, it's my name, Brynn @PennApps.com, and I will get back to you as soon as I can.